Good afternoon, everyone. Thank you for being with us. Just a reminder to remain muted until our question and answer at the end. And we have been given permission to record this lecture. So I'd like to introduce Pushpa Kalra, who will introduce our speaker for today.
Good afternoon. This is the last talk in this series on brain matters. Today we have Dr. Andrew Lu, who will talk to us on circadian clocks. Dr. Lu grew up in China and then got his Ph.D. in biochemistry from the University of Michigan in 2003. He went on to do postdoctoral research at the Scripps Research Institute in La Jolla, California. And in 2008, he was appointed as assistant professor at the University of Memphis. He was there until 2017 and was recruited to the University of Florida as associate professor in 2019. He is in the Department of Physiology and Functional Genomics at the College of Medicine here. And I would like to invite him now to talk about circadian clocks and what's involved in them. Andrew, you're on.
Sounds good. Thank you very much, Pushpa. Thank you for the invitation. It's my pleasure to talk to you guys about sleep and clock stuff. Can you guys hear me okay?
Yes.
Very good. All right, so I'm gonna be talking about circadian rhythms and clock. Sleep is regulated by the clock. So when we talk about sleep, we actually talk about the sleep-wake cycle. And this is intimately related to aging. So I will talk about two short stories. And one is mTOR nutrient sensing pathway. One is NF-κB inflammatory pathway. Both pathways regulates clock function and sleep. I will show some data. And these two pathways are related to aging, okay? So the very important point I would argue that sleep is still the best medicine, perhaps the best preventive medicine. It is important for a lot of purposes. Information processing, conserve energy. It's an active process. Sleep is actually an active process for a number of functions, including memory consolidation. It, in recent years, shown to be critical for what's called a lymphatic system, that is to flush out the toxins that's generated in our brain. Without this, people get really sick and causes neurological disorders. And circadian clock function and sleep affects nearly every single cell in the body, not just the brain. So it's essential for health. However, unfortunately, sleep insufficiency, including the timing, including the duration, perhaps more important, the quality of sleep. This insufficiency is very common in modern society, and it could be an underlying cause of diseases. I would argue also that sleep insufficiency represents a hugely missed opportunity that we should take advantage of in future. Unfortunately, older people tend not to sleep as well as younger people. However, there are exceptions. If someone in the audience say, well, I sleep very well, good for you. A lot of older people don't sleep as well as younger people. Again, 3 elements. One is timing, takes longer to go to sleep, and usually goes to bed earlier. And duration is a debating issue here. Are we older people sleep less or more? Certainly shorter at night, but because of the multiple naps for a lot of older people, the total duration may not change as much. The quality is certainly reduced. That includes the reduced deep sleep and spending less time on the critical phases of sleep. That includes the REM sleep and non-REM sleep, especially in the deep N3, N4 stages. And the shorter cycles, so our sleep consists of rapid eye movement and non-rapid eye movement cycles. The cycle goes shorter and shorter with aging. And certainly with reduced consolidation at night, this includes associated with sleep fragmentation, that is more frequent awakening and arousal, and more frequent transition to lighter sleep stages. So the questions in my lab we're asking in the context of circadian clocks and aging is to understand the effects of circadian sleep disruption on the brain health, on behavior, and age-related diseases. And an important aspect of my lab is to study the genetics, the molecular and cellular mechanisms of age-related dysfunction in the circadian system and in sleep. So I would argue everything is circadian, okay? Circa means about a day. That is to say that our endogenous clocks are about a day, not exactly 24 hours a day. But so look at the whole body, right? Our central clock, there's a master clock in the brain within the hypothalamus, which is the center for integration and regulates many, many important homeostatic functions. So the central clock is in the SCN in the hypothalamus. Virtually every cell in the body has a clock, and therefore every tissue, every organ has a clock system. What's important is the SCN organizes the whole body to become a coherent circadian timing system, okay? And once the system is organized, it regulates rhythmic outputs, including the central behavioral outputs, the sleep-wake cycle, the activity resting cycle, and the physiological and hormonal rhythms, including our body temperature and blood pressure. And these are important physiological outputs of the circadian rhythms. For example, body temperature is lower at night. If it's not lower at night, it's actually quite uncomfortable. Blood pressure is another very important output. Here, it relates to what's called nighttime hypertension. That's hugely missed opportunity. That is, hypertension patients tend to have go to the doctors to measure blood pressure during the day. And then we try to reduce the high blood pressure during the day. But what's really important is the nighttime blood pressure. So nighttime blood pressure is normally lower, okay? It is the high blood pressure during the nighttime that is the culprit or silent killer. Many other hormones, such as melatonin and cortisol, are these are the two prominent hormones that are rhythmically secreted and regulated by the clock system. Well, the clocks are about 24 hours a day, never really exactly 24. Therefore, it requires input, primarily the light-dark cycle, to entrain the SCN clock. The SCN clock then entrains the body. In addition to the light-dark cycle, our feeding fasting cycle, our activity resting cycle, also regulates the clock function, mostly in peripheral tissues. So in other words, the light-dark cycle entrains the master clock in the brain. The feeding fasting cycle and physical activity cycles tend to entrain the peripheral clocks, actually very effectively. Now, a critically important concept in circadian biology is the circadian clock provides an anticipatory mechanism. That is, before we wake up, our body prepares our body, including the body temperature, including the hormones, including the glucose levels. All these physiological processes are increased in preparation for the next day. So its anticipated function is critically important for the optimal function of our body. So this function is not just reactive. So, it's not like we wake up, we see the sun, then we prepare our body. The clock is there all the time to prepare our body. In order for the circadian timing system to function correctly, the whole system must be synchronized. So this is a very important concept that's called circadian synchrony. So this is basically summarizes what I mentioned in the previous slide. First of all, the light-dark cycle entrains the master clock in the brain. And then the SCN relays the time of day information. The SCN knows, okay, it's 8 o'clock in the morning. I need to tell everybody in the body, right, all the cells to say, okay, this is 8 o'clock. So what the SCN does is to relay the time of day information to the liver, the kidney, the adrenal gland, and so on and so forth via central output. The feeding fasting cycle has been recognized as a very important so-called like behavioral entraining mechanism to entrain peripheral clocks. The entraining signals include hormones and metabolites. And throughout today's lecture, we'll mention some of this. In addition, exercise, and when I mean exercise, I'm not talking about the voluntary activity. It's more like forced. You must have some kind of, heart rate goes up, and there's some hormones released, especially the sympathetic nervous system and the HPA axis, the cortisol, the adrenaline. These hormones must go up a little bit to become a potent, effective, resetting cues for the periphery. So, in summary, you have the light-dark cycle, and then the SCN tells everybody in the every cell in the body, every tissue organ in the body, what the local time is, and then the fasting and exercise activities entrain the peripheral clocks more effectively. So let's delve into the clock mechanism a little bit in a little bit more detail. So this is a cell, and this cell is engineered to express a luciferase. That's the protein that's expressed in the firefly. So we take that gene, we fuse it so that its expression in the cell is regulated by a circadian element. circadian mechanism. So this luciferase activities goes up and down, up and down. So from here to here is day 1, day 1, day 2, day 3, day 4. So this cell is doing a clock function, cell autonomously. It does not require the light dark cycle. It's endogenous. It's self-sustaining. So as you can see, this is gene expression essentially up and down. So that's the clock mechanism. That is, genes are activated and repressed. The mechanism of circadian clock function is activation of gene expression and repression. So more specifically, in mammals, in humans, in our cells, the activators are BML1 and clock. It binds to the E-box to activate transcription. And this regulates thousands of genes. A few of these outputs, the few of the targets of the activators can actually be transcribed, translated to repress the transcriptional activity of BML1 clock, to repress their own expression. So this cycle takes about 24 hours to complete, and therefore forming the mechanism of circadian rhythms. Here are some examples of clock or clock regulated genes. They go up day 1 and then day 2. This is another gene, this is another gene. So this mechanism underlines a rhythmic genome. About 50% of all genes, we have about 30,000 genes in our genome. About 50% of all genes cycle somewhere in the body. For example, it's about 3,000 genes that are rhythmically expressed in the liver and 2,000 in the kidney, okay? And these genes are expressed like here, like this, are expressed at different times of day. There's several waves. Some genes are highly expressed in the morning, the others in the evening, the others at night. And these genes are largely tissue specific. That is to say that these 3,000 genes in the liver that are rhythmically expressed in the liver are largely different from those in the kidney. Now, so aging is associated with circadian decline. So that's shown in animals. So whether we knock out, we delete a core clock gene to make the clock go away, or we use environmental approaches called chronic jet lag. That is with phase advance. You know, every few days you let them travel 6 hours in advance. So chronic experimental chronic jet lag. All these conditions causes accelerated aging. The mice die early. In humans, overall, what you see, the pattern here, the most important pattern here is all the rhythms are dampened. So instead of having a high amplitude physical activity, sleep-wake cycle, temperature cycle, hormones, the overall amplitude, that is the differential between the peak and trough, are reduced. So translating to the behavioral, sleep or activity rest cycle, you see with aging, our day, unlike teenagers, our day is okay, our night is okay, but it's not beautiful day and a wonderful night like the younger people. But a good thing is the happiness. Older people actually are happier. This is actually a very good thing. The happiness across the lifespan actually takes a U shape form. At about 40, 50, you have a midlife dip here. But once you jump out of the dip, actually people are happier. So it's actually a very good thing. So in summary, clock is important. It regulates sleep. It regulates most, if not all, the major aspects of physiology, metabolism, and behavior. Older people tend to not sleep as well as younger people. However, older people are happier in general. Now the clock genes, that's the main major research program in my lab, that is to identify genes that regulate the clock. And in recent years, we start to study more on the sleep. If these genes, the pathways can regulate the clock function, they most time regulate the sleep. So this study is a one of the kind, was completed about 10 years ago. What we did there back then was we have a clock, a core mechanism. You have activation, you have repression. You have about 10 genes making up the clock, the core clock mechanisms. And then the question was, are there other genes in the entire genome, in the human genome, that regulate the clock function? Are there additional clock genes? Are there clock modifiers? So we did a genome-wide screening. The question was, we knocked down every single gene in the human genome to ask, does it have any impact on the clock function? So showing here are two examples. That is, you see the black trace is the control. The clock is running about 24 hours a day, okay? But if you knock out and knock down, reduce the level of a particular gene expression, you see the clock runs much faster. Instead of 24 hours a day, this is about 21 hours a day. And the other genes, when knocked down, can lengthen the clock, the speed of the period length, that is their endogenous day. Two examples I'm going to talk a little bit about today are mTOR and NF-kappa B. So mTOR basically links nutrient and energy sensing or metabolism to circadian and sleep homeostasis. So what is mTOR? Well, mTOR is one of the top, if you do a Google search, it will be one of the top 5 most studied genes ever, okay? What it does is it regulates protein synthesis, cell growth, and proliferation in response to higher energy. So there's a lot of glucose, a lot of amino acids, and in response also to growth factors. So there's enough energy and there's a signal to grow, and this pathway is initiated to promote protein synthesis, nucleotide synthesis, fatty acid synthesis for growth. mTOR is known, perhaps most known, as a target of a drug called rapamycin. So it's a mechanistic target of rapamycin. Rapamycin is commonly used as an immunosuppressant in kidney transplantation. It has been shown to extend lifespan in all the model organisms studied, including mice, including Drosophila, including C. elegans, and so on. Okay, and in addition to improved lifespan, it actually also helps health span, that is, improves aging-related chronic conditions, if healthy aging, if you will. Okay, so I studied circadian clock function. What we did, this is one representative data to show that when we put the rapamycin in clock cells, the clock runs very, very slowly. Their day is about 27 hours per day, okay? And the mechanism, take a long story, takes several years for us to finish this project. The long story short is mTOR regulates the protein expression of one of the core clock components, primarily Cry1. Cry1 is the repressor of the activator of this negative feedback mechanism. So mTOR activates, promotes Cry1 translation and abundance. Rapamycin inhibits that process. That's in cells, and similar in the master clock in the SCN. We can actually dissect the SCN out from mice and culture the SCN ex vivo. That's called ex vivo. And we can study gene effects. We can add drugs onto the SCN to see whether the SCN is affected by certain perturbations. This is mice. What we did here is genetically, we delete. The mTOR is an essential gene. We cannot delete mTOR, but we can delete one copy of it. Deleting one copy, if we will, the loss of function of mTOR makes the day in mice longer. The experiment may want to explain a little bit how the experiment is done. Is we put mice on a running wheel. It's a running wheel cage. Mice like to run. They run 5 miles per night. Okay, that's voluntary. If we force mice to run, they start to do different things. That is, if they are forced to run, they actually can entrain the peripheral clocks. But voluntary doesn't do that. So we put mice on the running wheel. They start to run on the wheel. They sleep. They run on the wheel and they sleep. Okay, and then we release them into constant darkness, no light. And we call that free running. The clock is running by itself. It's not reset by the light dark cycle. The mice get up 30 minutes earlier per day. If we put humans in constant darkness, we tend to delay half an hour. We're not getting up earlier, we're getting up a little later. Mice gets up a little earlier every day. So that's the way to measure the endogenous clock function, the SCN. So as I said earlier, in constant darkness, as well as in constant light, the endogenous clock is slower. A study by a colleague published around the same time showing that rapamycin, which is the inhibitor of mTOR, can lengthen the behavioral rhythms, and that the behavioral data is consistent with our behavioral data. It's also consistent with our cell-based and SCN-based experiments. Now, mTOR function in the sleep-wake cycle, we got some insights from patients. So this is a very new, relatively new disease discovered in 2013. And in collaboration with my collaborators, we have identified more than half of all the more than 60 mutations in humans. This is mostly like a autism-like disorder. The clinical and cognitive phenotypes include intellectual disability, developmental delay, megalencephaly, and seizures. Right? In addition to that, what is being largely ignored in medicine, especially this particular patients, is their homeostatic functions. In this particular case, it's sleep. The sleep problems not only impact the children, the patients themselves, but also the caregivers. In particular, these kids need care at all times. If the kids are not sleeping, the caregivers, the parents, are not sleeping either. So what we show, this is assay to show, what happens with the mutation, the protein itself. What we show, this is just take a look at this mutation. This is the deletion mutation. We know who the patient is, right? We actually treated these patients. We show that under basal conditions, that is, we're not activating purposely the mTOR. We're reducing the basal activity to basal levels, and we compare the mutant mTOR with the wild type mTOR. We show the mutant mTOR is much more active than the wild type, and that's summarized in this chart here. Now, what's really important here is all these mutations are not the same, and that is critical for us because we tend to think all these mutations are the same, and we treat the patients in a similar way using the similar amount of rapamycin. That's not the case. And in addition to the differences in their intrinsic activity, we also show that these mutations respond to rapamycin in a different manner. So that's the caution for us going forward. When we treat patients, we need to look at the patients on the individual basis. So now I don't have charts or beautiful data sets. Just in words describe what we did with the patients. Now, sleep problems are common in SKS, Smith-Kingsmore Syndrome patients. In addition to sleep problems, they also have feeding behavior problems, hyperphagia, body temperature control problems. Some kids actually feel hot even in the winter. And they have all of this, okay, indicates a dysfunction in the hypothalamus and in the SCN. That's where the homeostatic controls are. With one particular one patient, this is deletion mutation that I showed earlier, that in the previous slide, this mutation is highly active. They go to bed early and followed by a very irregular sleep cycle. And when we treat them with low-dose rapamycin, when we treat it with high-dose rapamycin, we caused the sleep timing opposite direction. Before treatment, they go to bed early. After high dosage treatment, they go to bed very late. Instead of 8, 9 o'clock, now it's 2 o'clock in the morning. So, and that's the first time we saw, we really know the SCN is affected. So we actually were able to adjust the dosage to make the kid sleep in the relatively normal pattern. So optimized rapamycin dosage corrected the sleep-wake cycle and also improved clinical symptoms. So from these patient treatment finding, we see the sleep-wake cycle as a biomarker, okay? Not only for sleep, but also for systemic homeostasis. Now, as a basic scientist with the Ph.D. behind my name, it's really gratifying when we hear the mother of the kid, sending us a letter. This was a year ago. This treatment continues till today, and the sleep is significantly improved. What she told us is this has given our family the gift at that time, 30 days of good sleep in a row, with the hope of more to come. The improved sleep over many consecutive weeks can only help him experience an improved quality of life that had previously been unavailable for him for the past 17 years. In other words, for the first time in 17 years, he and his family can have a good sleep. So we have generated CRISPR mutant mice to try to study these mice, the mechanisms and how they work, and can they inform treatment options. Now this is reminds me it's almost done. I need to sleep, and Albert Einstein sleeps a lot also. Now NF-κB, it's inflammation and clock function. What is NF-κB? Well, whether you know it or not, if you're a scientist or biologist, you will know. If you don't, well, all this listed here will cause NF-κB activation and inflammation. Now, the activation of, in addition to its normal function, that is to repair, constitutively overexpression or overactivation is a very primary cause of a number of diseases. And here, of particular relevance in human normal aging, there is what's called low-grade inflammation termed as inflammaging, okay? the NF-copy activation, the pathway, is a key driver for inflammation. That is low-grade inflammation during the normal aging process. We show that NF-copy, when we introduce it into cells, it changes the clock function, both in cells and in the SCN. The mechanism is NF-copy actually directly hijacking the clock mechanism by directly binding to the clock activators and compete with the repression by these factors. This is directly hijacking the clock mechanism. It changes animal behavior. I'm not going to go to the detail. It also changes sleep. Here, what's shown, I'm going to use this to introduce what this means. When we activate NF-copy genetically, we introduce the transgene into mice, and we remove the stopper to activate the pathway. When we activate the pathway, what we see is the sleep quality. What's shown here is the two categories. One is called short bout-length sleep. That is, the REM non-REM cycle is short. And the other on the right-hand side is long. So the short sleep cycle is bad. The long sleep cycle is good. So when NF-copy is activated, you have more time spent in the short sleep bout-length sleep, bad sleep, but spend significantly less time on the good sleep. So that's the rapid cycles is considered sleep fragmentation. So NF-copy causes sleep fragmentation. Brief introduction of the microglia, which is the innate immunity cell in the brain. What we show, this is published last year, showing that in old mice, okay, these markers for microglia function as well as exercise function is dramatically increased. And we showed in our own studies, but more specifically in the hypothalamus where the SCN is, all the microglia markers are significantly higher. So this is like a silent killer. These are normal mice. We're not doing any stress, to the mice. So normally these activities are higher. And we actually did, sleep assay with these mice. We show indeed the older mice spend much less time in the good sleep, sleep in the good sleep category. So this sleep data is consistent with our NF-copy genetic data, also consistent with the microglia shifting high gear in old mice. So this is an emerging research field that is to increasingly recognize the role of the inflammation in the hypothalamus, particularly in our case in the SCN, in normal aging, as well as in aging-associated diseases. Now, I want to come back to I'm sure everybody is familiar with a term called caloric restriction. That is to say, instead of eating 100% or 150%, we eat 70%. That is very good for lifespan and health span. But we don't want to eat 70%. 70 is not very happy, okay? But overall, 70% reduction, 30% reduction, eating 70% is the most robust anti-aging innovation, both for longevity and for health. Now, this is a complicated diagram, but I want to make it simple. That is, caloric restriction causes longevity. How does it do that? At least three important master regulators. And these regulators, as we show, I didn't talk about this. This is done by other labs. That these three master regulators regulate the clock function. Caloric restriction can Activate SIRT1 is another very important anti-aging gene or pathway. Activates SIRT1, represses mTOR, and represses NF-κB inflammation, leading to longer lifespan. Now, as I mentioned earlier, we don't want to eat 70%. That's just painful. But happily, so a different study a few years ago showed, well, let's see caloric restriction, 70%. In mice, what this group showed that if you give mice 70% of food, they actually eat the food within the 2 hours, the first 2 hours, gone, food is gone. Okay, that is actually, conceptually speaking, similar to what's called time-restricted feeding, where food is available within the active field time, that is 8 to 12 hours active time. No food, actually mice tend to eat a lot. In humans, studies show in humans, 18 hours out of the 24-hour day, people eat food. The high-fat diet causes obesity, but high-fat diet with time-restricted feeding, that is no food when there's during the sleep period, actually reduces the body weight. And in addition to body weight, all other good factors are increased under time-restricted feeding. All the bad stuff is decreased. So there's benefit for the time-restricted feeding. So this is like a circadian element, right? So we're not reducing the caloric restriction. We're not reducing the calorie intake, but we just restrict the food during the daytime in humans and during the nighttime in mice. So this is a novel concept. In summary, right? So we talk about nutrient excess, overnutrition is not good for aging because it activates mTOR, NF-κB, which adversely affect the circadian rhythms and adversely affect sleep-wake cycle. Caloric restriction is very good for anti-aging, but it's painful. It's just not sustainable. There's so much good food out there that we just have to enjoy. Now, let's do time-restricted feeding. Instead of just cause pain not eating, let's do time-restricted feeding. That is, we eat within that 8 hours or 10 hours or 12 hours, but don't eat for another 12 hours minimum. Time-restricted feeding, and eat it at the right time, not midnight burger. Midnight burger is bad. In addition to time-restricted feeding, we need to conform to the light-dark cycle and to follow our sleep-wake cycles. In addition, as I mentioned earlier, exercise that is a little bit like forced physical activity is also good for the circadian cycle. Unfortunately, our modern lifestyle causes a lot of problems. These are what's called asynchronous cues. Light at night, that is not just the light at night. All the electronic devices are considered that contributes to light pollution. Sleep insufficiency is another problem. No physical activity or less or mistimed physical activity is probably not good either. Unhealthy diet, certainly not good. Mistimed feeding is not good either. The physical activity and when to perform physical activity, and these are actively pursued research areas today. And so these are not good. In summary, and this is more like conceptual, if you will, given the wide-ranging health benefits, minimal cost, no side effects, and accessibility, all of these regimens, okay, present to us an attractive, that's a keyword, non-pharmacological treatment and preventive option. We're not taking rapamycin, we're not taking metformin, we're not taking resveratrol. We're using this non-pharmacological approach to enhance sleep function, to improve sleep-wake homeostasis, and ultimately improve aging physiology, which offers a benefit to maintain mental and physical health and overall well-being. So this is more or less a summary, okay? Despite decades of hundreds of years of study about the sleep, we actually don't know where controls sleep, what genes controls sleep. We know where the clock is. That's in the SCN in every single cell. We know the genes that regulate the clock. Hey, maybe we should control the clock to increase our health. So control what we can control, that's the clock. In summary, circadian rhythms are important, and the two pathways we're studying, one is nutrient sensing, the other is inflammation. By modulating their activities to optimal, when I say optimal, I'm talking about two things. One is their level, one is their level at the circadian dimension is supposed high during the day, low at night with respect to mTOR. And it's not just the level, it's circadian. All of this enhances circadian sleep homeostasis and promotes healthy aging. In future studies, we're using these two pathways to study how they regulate sleep and how that might improve cognitive functions in aging and aging-related pathologies. And finally, I want to thank a few people who are involved in this, the data I presented. Chida Ramanathan was a previous postdoc. He contributed to the mTOR study, and Yang contributed to the NF-CoB studies. And these three, Destino Hongzhi and Mahari Andy Morris, contributed to the behavioral studies of the NF-CoB project. I want to thank also the funding sources, the NIH, for, in particular, the foundation support from the SKS Foundation Research Fund. And that's it. I want to thank you all very much. I may take some of your questions.
You want to take down your screen?
Yep.
Okay, thank you. We do have some questions from chat. It says, first one says, could you define what is meant by gene expression?
Gene expression. In general, right, the genes are all the genes are in our genome. They're in every single cell, and the expression of these genes are regulated so that different cells in different tissues and organs express different genes. And in the context of circadian clock, it not only regulates where it's expressed, but also when it's expressed. Does that answer your question? And the regulation is more like the DNA, right? The DNA is the blueprint. That the structure of the DNA was one of the prominent Nobel laureates who discovered all the DNA is double helix. But that's the blueprint. In order to express itself, it needs to be transcribed into messenger RNAs, which then serve as a template to translate into proteins, which then perform functions. I hope I answered your question in the right.
So do you mean production of proteins by the gene as the output of the expression?
Yes, that's what I mean.
Okay, Elette, while you're there, do you want to ask your other question?
No.
Okay. Shirley Bloodworth, go ahead.
This is fascinating. Thank you very much. It's hard to understand a lot of it, but it's fascinating. Many older people who have sleep apnea and lung problems have a drop in oxygen overnight. How does that impact the clock, and how does that impact brain function and sleep proficiency?
Yeah, it does. We know this low oxygen is called hypoxia. Okay, hypoxia is we know hypoxia, the gene that regulates or the pathway that regulates hypoxia called HIF. HIF actually was a Nobel Prize winner last year or the year last year. So we, not my group, but a friend of mine and three groups show that HIF directly regulate one of the core clock genes called PER2. Okay, and that's one. That is, hypoxia impacts clock function and most likely sleep. I'm not sure if people have done sleep. But since you talk about hypoxia, I want you to relay this information. That is oxygen treatment. Oxygen treatment can improve aging physiology. Recent studies show there are 2 clinics in the world. Okay, one is in Israel, one is in the villages. I don't know how they do it. I think they put you on the oxygen tank, maybe just you breathe for 15 minutes. That seems to do a magic stuff. I don't know how much you have to pay, but it's in the villages. Oxygen treatment.
Well, oxygen treatment is necessary, and CPAP treatment is necessary for some people to maintain oxygen levels. So we are doing some treatments. I don't know if it's keeping us young or not.
Yeah. So yeah, the insomnia related to a very good friend of mine in Cincinnati Children's. He's a Ph.D., MD, Ph.D. So he's actually my collaborator in the sleep studies in humans. His main expertise is insomnia, but in kids. Yeah, it's very, it's heartbreaking when he tells me all these stories about kids.
What kind of subjects are you looking for in your research here?
We mainly study clocks, so genes to regulate the clock. But what's I want to expand a little bit. There's the clock. We know there's these genes there, but what we more and more realizing is it's not just the clock genes. Actually, all these processes, all these physiological processes, we go out, have a run, we know the clock can be monitored, can be modified by it. So we're studying all these cellular and physiological processes that can change, for better or for worse, the clock function. As one of the main outputs of the clock function, the sleep-wake cycle. So my research is more on the clock function, sleep-wake cycle, mostly in the brain. In recent years, we're studying more of the age-related decline. That's because the 2 pathways we're studying, as I alluded in the talk, these pathways actually are major pathways for regulating aging. So the whole thing came together, say, well, these pathways regulate clock function and aging. Perhaps these pathways actually regulate aging through regulating clock function. Suddenly we have a handle, about aging, which is a long process. Now we study, hey, let's not talk about 80 years. Let's talk about today, All
right, we have a few more questions from chat. One says, is taking melatonin at bedtime effective in improving sleep quality, quantity?
Yeah, I want to be careful when answering your question. I'm not a physician, so I don't offer, but I take melatonin every day, every night. With melatonin, 50% of the population are not responsive to melatonin, perhaps because of lack of sufficient melatonin receptors in the sleep centers. But I'm very, very fortunate. I'm very sensitive to melatonin. I use 1/10 of the amount that's recommended by physicians. So I basically had to buy the lowest, dosage tablets. I actually cut into pieces so that I only use like 0.3 milligram instead of 3 milligram. So so to answer your question, is it effective? But 50% of people are not, not very responsive to melatonin.
Sounds like a placebo effect with you, Andrew.
All right, the next question says, does practiced meditation for at least 1 hour work as well as sleep?
So not my study area. However, there are several prominent labs. One in particular at UCLA, Irvin's lab showed that meditation is single most effective form in improving sleep. Okay, so that's really in the past maybe 10, 20 years that they did this. Yoga, meditation. He's written several papers, including reviews. It's very impressive. So I actually heard his talk once. I'm rather convinced. So I actually told myself to do meditation. Basically, what I do is I listen to my breathing. Seems to be effective.
Okay.
But there are other important things we can do. I think all of us can do. That is, we're in Florida, we're so lucky to have a lot of sunshine. So we can expose to sunshine during the day. Okay, our brain, our eyes actually can remember the history of light. That is, bright light during the day can make the light at night less effective. So light at night is no good. It's not good. We know that, right? But if a brighter exposure to light during the day can make the adverse light effect at night less. That's one thing. Two is regular. So to maintain regular routine, I'm just talking this on paper. I can't do that. Regularly you should go to bed at 10 o'clock. Regularly you should not watch movies on Netflix. I tell you this because I'm implying I actually do that myself. So it's actually bad, but you can't help. So maintain a good regular routine is very good. And the other thing in the context of circadian clocks is the temperature cycle, right? So most of the time light equals high temperature and night equals lower temperature. So we should maintain a little bit lower temperature, room temperature, to help sleep. These are some of the common things. Melatonin, I use it every day.
All right, another question says, could you speak of high blood pressure during sleep? How would we know?
I'm sorry, how would we know?
High blood pressure during sleep.
Okay. Yeah. This is a very, very active research. We physicians, we actually had I had a meeting with 2 prominent hypertension researchers in Houston several years ago. In the group, there is a physician who is hypertensive, who is taking beta blockers to reduce hypertension. He, as a prominent doctor, does not know that you need to take medication at night before bed. Okay, so how do we know? How do we measure hypertension? Unfortunately, well, do you guys have Fitbit?
Some do.
Yeah, some do. So you can actually monitor your hypertension, blood pressure, sleep, even the sleep quality using these devices. Okay, and you can actually monitor this month versus next month in real time. That's one thing we can do. In doctor's office, unfortunately, I actually consulted with a physician, cardiovascular specialist, a friend of mine I said, I want to be monitored, called longitudinal recordings. But he told me, yeah, you have to be sick enough to do that, to be covered. So we cannot just go to the doctor's office. So say, well, I want this called ambulatory blood pressure monitoring system. We cannot do that. But what I do want to say is the blood pressure, the high blood pressure, the hypertension called nighttime hypertension, or known as dippers or non-dippers. So if your blood pressure is not dipping at 2 o'clock in the morning, that is the killer. That is the culprit. That is a hugely missed opportunity in medicine. That we should really be treating the hypertension, nighttime hypertension, that's 2 o'clock. So I think if you're all happy people now, so buy a Fitbit or what's called a watch, the Apple Watch. I think they can monitor the blood pressure. I'm not so sure about the sleep. I think Fitbit can monitor sleep. And these are important, basic functions of our body that this technology can allow us to actually have access to. Daily monitoring, it's kind of important to know our health in real time.
We have another question from Mary on the chat box. It says, you mentioned controlling all these aspects of our circadian rhythms, eating, exercise, sleep. What do we do to discern what times of the day or night for doing these that are appropriate for us?
Okay, let's start with sleep. Try not to sleep during the day. Try to sleep at night. Although it tends to be difficult. I'm telling you, I struggle with sleep. I'm not a morning person, so I'm awful in the morning. I don't wake up until 10, so that's a problem. So that actually relates to exercise. What time to exercise? If I knock my wall, the person in that room is a muscle physiologist. Her lab and my lab are together. So she's studying physical activity. As to what and when, okay, so our suggestion at this point is, for me, I'm not a morning person. I should not run 3 miles in the morning. For me, it's not I should not run. I cannot run in the morning, okay? So if you are a morning person, it's okay. But morning tend to, from the circadian point of view, morning tend to be a crisis time where all the blood pressure is going up, the autonomic nervous system goes up, glucose go up. All these different processes are going up to prepare us. But if we're not ready, that's where the problem can go awry. In fact, don't quote me, I think it's probably 70% of the CVDs, the cardiovascular stroke patients, have problems in the morning time. With respect to stroke, the document says it's about 9 o'clock, but stroke, the stroke occurs actually prior to 9 o'clock because actually it takes time for them to go to the hospital. So morning is, for me, it's so bad. And that's because my clock runs very, very, very slow. Okay, I don't feel like sleepy until, that's why I use melatonin. But for those you who get up in the morning, feel energetic, why not go running? So sleep, exercise, eating. So eating, I would recommend 10 hours. Within that 10 hours of our daytime, eat during that period of time, giving yourself about at least 12 hours, if not more, maybe 16 hours of so-called fasting. That tend to sensitize our important metabolic processes such as insulin sensitivity.
Holly Hunt has asked for a citation on meditation that you mentioned on meditation research. Could you send it to me and then I can pass it on?
Yes, yes. Let me. I will send those references to you.
Yeah.
Yeah, and then yeah.
I might add that this just leads me to the next series of talks that I'm arranging. And one of the talks in that series on alternative medicine will be on meditation in the summer. So all those interested in that. Andrew, I have a question. I myself worked on the hypothalamus and the SCN, but our research was on neuropeptides. The neuropeptides that induce sleep that were discovered were orexin, delta sleep-inducing peptide. Have you seen any or studied any connection between these genes and those sleep-inducing peptides that were known before we knew what genes, what these genes were identified?
Yeah, for your microphone actually makes the whole echoing. I can't really hear it very well, but I think your question is the genes that encode peptides, neuropeptides.
The neuropeptides that are known to induce sleep.
Neuropeptides that are known to induce sleep.
Yes.
Not a whole lot. We know several that are involved in sleep. Neuropeptide 1, Y, NPY, that's actually a feeding, appetite regulator. And the other one is hypocretin or orexin. That's for wakening. That's right. So I think without answering the question, specifically, the problem with sleep is we don't know where the sleep center is. We actually don't know how many nuclei in the brain, including the hypothalamus, that regulate sleep. And I might want to add one more, that is, we don't know how many genes regulate sleep. That's hugely challenging.
So these genes that you have been studying, they are not related to the neuropeptide function. That was my question.
They do.
Is there a connection between these genes and those neuropeptides that induce sleep? Or are there other genes that could be regulated? Because I'm very excited to know what the genes are in the SCN, circadian rhythms. But we had worked on neuropeptides, and NPY was our peptide, orexins, and creatine. So that's why I'm looking for the connection.
Yeah. So the SCN is very small. Okay, so it's about 20,000 cells in the SCN. So I think people have done this calculation to show that it's not necessarily what the SCN secretes. It's actually what the SCN targets, the other nuclei in the brain, in the hypothalamus, as well as other peripheral, regions. So I will not be shocked if people show that leptin, insulin, these hormones actually regulate sleep because they actually target the hypothalamus.
Right.
Yeah.
There's one other question in the chat box that says, do some people need less sleep?
Absolutely. I had the fortune to work with two prominent, they are all National Academy members. And one is my PhD postdoc advisor. One is, I worked in the company for 2 years, and he's the boss. So both of them sleep 4 to 6 hours per day. They're totally okay. They're called short sleepers. Two groups in UC San Francisco are targeting these people. They're trying to identify short sleepers, 4, 5, 6 hours per day, totally functional. They actually identified identifying genes that are responsible for the short sleepers. The short answer to your question, they are these lucky ones that sleep less and functional, and I'm not one of them.
Are there any questions here in the room? Anybody? So come up here to the microphone, please. Who does? Question? No. Okay. Okay. If there aren't any more questions, then I would like to thank you, Andrew, for taking the time to present this very new field to us. It's fascinating, and I think it's been very interesting to all our audience too. And I would like to talk to you sometime about the specifics of the research. Okay, thank you very much. And this concludes the series on brain matters for this particular series of the ILR. This was the last lecture. Stay tuned for the next one in summer, alternative medicine. Thank you all.
Thanks to you all very much for this opportunity. Good to see your faces.
Bye.
Thank you. Okay, bye-bye.
Thank you, everyone.